Good morning. If I could have your attention. Thank you so much. Welcome to the 27th NEURON Conference here hosted at Quinnipiac University, the lovely North Haven campus. We're super glad that you're here, especially considering all of the weather issues that we've had. I'm going to run through just a couple of slides to let you know, one, that you're here at the Frank Netter Medical School. And so make sure you check it out. We have some of Netter's drawings hanging on the wall as you go around the oval here, or around the donut. So make sure to check those out. I'm Dr. Todd Ahern. I'm an assistant professor in the Department of Psychology in the Behavioral Neuroscience program here at QU. And this is Dr. Betts. She'll be speaking to you in just a minute. We're the local organizers, and so thank you in advance for accommodating our delay and everything else. I think that it's going to be a great event. Okay. If you want to, it'd be great if you could tweet about this conference. So there's the hashtag if you have it. So it's a good way of kind of getting it out there. You can also check out our Facebook page. There are photos from last year. And in order to encourage more photos, we're going to ask you to all take photos, and we've got a site that you can send those to. Okay?
There's a slide.
Yep. So if you want, you can also check out our YouTube channel. The keynote address will be recorded today and will eventually be posted there. So if any of your colleagues weren't able to make it, and we're really interested in seeing her talk, it will be available on the QU YouTube site that you can find through here. Okay. You can also find ones from last year. So Dr. Eisch last year presented, and so you can find out her talk there. If you are going to take pictures, all you have to do is email them to this email address, and they'll all get clustered together, and we'll be able to show that this was a vibrant conference despite the snow. Just to let you know, you can see here that over the last several years, NEURON has grown. We're a little bit smaller this year because of the growth, but we think it's a real testament to all of you that you were willing to come. It's been a great conference, and you can see that in terms of number of posters, we had 96 that were originally registered this year, and then we had upwards of 350 original registrants. So we're very excited by the growth of this conference, and we're glad that you're here. We also had some data from previous years in terms of how engaged students have been and faculty have been, and that they want to come back. So we're hoping that you'll have that similar experience this year, and we'll be sending out a feedback survey. Either you can use the QR code, there are a couple of them around, that you can fill out the evaluation survey to let us know how we're doing, or we'll send it out in an email after the conference. Okay? We also have a bunch of different institutions represented from all over the Northeast, and so you can see there are a bunch listed here. Yours is probably up there. And so we have 41 registered for this year. And you can see the geographical reach of this conference. So we were pretty clustered in New England. We even had some registered from Maryland this year. Not sure if they made it or not. Is anyone here from Maryland? No? All right. So with that, I just want to introduce again Dr. Betts. She was the chair of the organizing committee. And I want to point out that there is a program, but as you notice that because of the hour delay, there's an insert so you can follow along there. Or you can use the electronic version, which has been updated, and the short URL is there. Okay, so welcome and thank you for coming.
Thank you, Dr. Ahern. I'm just going to take a couple moments to tell us about the poster awards that are available today at NEURON. I am looking for faculty, postdoc, or research scientists who are interested in judging these posters. Our poster coordinator from Skidmore did not make it down, so we have a little bit of a void there. So please seek me out if you can. So the first award will actually go, this is at the end of the conference, to a faculty member to honor Mary Erskine. Then we have two different undergraduate and graduate poster awards. The first one is the Susan Tiemann Award, so we'll have one for undergraduate and one for graduate, and then the Fry Pioneering Research Award. So for the Fry Pioneering Research Award, this is going to be honoring research that you if you're of a minority group, but you need to opt into this particular program. So a judge will not ask you if you are eligible for this award. So if you'd like to see what the criteria would be, please download a QR reader. You'll see this QR code around the conference. Just scan it, and you'll be asked a series of questions. This is also how you'll give us our feedback. So again, you'll see 2 different QR readers, so please just take a moment. And if you have any trouble downloading a QR reader, just seek one of the conference helpers out and we can help you with that. Okay, so it is now my distinct pleasure to introduce Dr. Rajita Sinha. So Dr. Sinha received her BA from Delhi University and her PhD in biological psychology from Oklahoma University Health Center. She went on to conduct postdoctoral research at Yale, first in the psychology department and then in the Department of Psychiatry. Since then, she's become a Foundations Fund Professor of Psychiatry, Neurobiology, and of the Child Study Center at Yale. She's also the chief of section of psychiatry and the deputy director of the interdisciplinary research at the Yale Center for Clinical Investigation. She's the founding director of the Yale Stress Center. This was established in 2007 when she led an interdisciplinary group of basic and clinical scientists to set up an NIH Common Fund, which was supported by interdisciplinary research for a consortium on stress, self-control, addictive behaviors, for example, nicotine, smoking, alcohol abuse, and overeating. So her own research, she has over 200 peer-reviewed publications and has made discoveries on stress mechanisms that link the maladaptive behaviors and poor health outcomes, and on developing and validating novel stress reduction strategies. She and her colleagues use molecular, genetic, pharmacological, imaging, and other approaches to link the neurobiology of stress to emotional regulation. Dr. Sinha's lab is funded by NIMH, National Institute of Diabetes, Digestive and Kidney Diseases, National Institute on Alcohol Abuse and Alcoholism, National Institute of Drug Abuse, and many, many others. Through these organizations, she's received several awards, several types of grants, R01s, R21s, training grants, T32s, and through these grants, she's fostered and mentored countless students. She's also established a unique evidence-supported integrative brain, mind, body clinical care service to improve health and well-being. As a direct result of the science disseminated from the center, in 2012, the Yale Stress Center included a new initiative called the Yale Stress Center Clinical Services. This center focuses on an integrative whole mind-body healthcare. It includes services such as acupuncture, endocrine function assessment, biofeedback, mindfulness, and much more. Dr. Sinha has served on many NIH special emphasis panels, review committees, and workshops. She's presented at numerous national and international conferences, and her work is widely cited. As Quinnipiac grows its own commitment to neuroscience and seeks to attract new students, I particularly admire Dr. Sinha's unique ability to make science accessible to a wider audience. This is evidenced by her role in large training grants, countless speaking opportunities such as today, and a recent article in the Scientific American titled, "This is Your Brain in Meltdown." It's a great article, so if you haven't checked it out, look at it. She has served on several scientific advisory panels for the Science of Behavior Change program at the NIH Common Fund. She's currently on the National Advisory Council of the National Institute of Alcohol Abuse and Alcoholism at NIH. She's a very sought-after speaker on stress and the effects of brain, health, and behaviors, and she conducts workshops, lectures, and retreats on stress management, self-care for the stressed professional and for senior executives, on all ways to enhance your work-life balance. Please help me in welcoming Dr. Sinha to the 27th NEURON Conference.
Thank you so much, Dr. Betts. I want to thank you for inviting me, and thank the NEURON Conference for having me. And thank you all for heading out on this treacherous morning. Hopefully everybody's cars did okay, and you're here, so I'm sure it did. It's a real pleasure to be here. I love going out and talking about the brain and mind and stress, particularly because of really how rampant it is in our world today. And I hope to share some of that with you, and then really talk about how we perceive it and handle it as humans, and what are the challenges, and then also the hopes and the optimism. So with that, here's my title. I'm going to talk about, just sort of give a sense of what I mean when I talk about stress, the types of stress, and which are the ones that are sticky and affect us in a deleterious way. We'll go into the brain a little bit, talk about its effects on emotion and cognition, take a step back and talk about stress doesn't just happen today and yesterday, but really can start in childhood, and particularly its effects in development, and how that sets the stage for risk for common chronic diseases, its effects on behavior and health, and then really the hope that there is from neuroscience and from the clinical studies that are going on that are telling us about how dynamic the brain is, and that there's hope to change, perhaps, and reverse some of the effects of stress, which is very exciting to all of us who are studying it. So with that, let me just start with talking about stress and what it means and what it is. We all know what it is. If I took a poll right now, you all would give me some examples of what you found stressful. And I'm going to ask you to be thinking about that. So there's work stress. There's financial stress. We've had a lot of that in the last 5 years, 7 years since the meltdown. There's parenting stress. If you're a working parent at multiple demands, perhaps you might get really stressed out with handling those multiple demands. There's relationship stress. Divorce rates have sort of stabilized, but one more worrisome trend is that less people are getting married. And that could be something related to not wanting to get into the stressful part of relationships and marriages. So clearly there's relationship stress. There's family stress, and particularly children are in the midst of that. Chronic diseases rates are going up. They are rising in multiple facets. We'll talk about that. But really that creates a lot of stress in terms of handling one's own limitation, facing the challenges of treatments and all of that. And then there's life event stress, like yesterday, snow and Sandy, Hurricane Sandy, and various other life events, unexpected, unpredictable stressors that can be uncontrollable, and how we approach that can be challenging. And so needless to say, this is important because there are high levels of stress in the U.S. The American Psychological Association has been doing yearly surveys. If you go to their website, you will actually see the data from every year. And so, for example, they have shown that over half of the American public will report being pretty stressed. And the more concerning thing is that children, over a third of children will say they're very stressed and don't know what to do about the problems that they have. Even more, as we dig deeper, even more concerning are that way more than 1 in 3 folks have had 2 or more adverse childhood experiences, and those are listed here in terms of what I mean by childhood experiences. We'll talk about adverse childhood experiences as well, but substance abuse or mental illness in the home, verbal, physical, and sexual abuse, family member in prison, witnessing domestic violence. These are sort of the target questions that the Center for Disease Control actually monitors in terms of adverse childhood experiences, and there's quite a bit of data on that. Almost half of U.S. men and women report being a victim of psychological aggression, not even counting physical aggression, which is out there, of course, by an intimate partner or by somebody they know. And then lifetime prevalence of anxiety disorders, these are the DSM-5 anxiety disorders or acute stress disorders, are close to 1 in 3. So this would be a direct translation of stressors, although you will see from the talk that there are other chronic diseases that are directly affected by stress. So what do I mean by stress when I use the word stress? Any challenging, threatening, overwhelming event or stimuli that increases a stress response to regain homeostasis. You're going to see that we all are hardwired to have a stress response. It's there as humans and mammals and other species. We all have a biological stress system to alert us when danger is coming our way, when challenging things are coming our way, and essentially to protect us and help us survive. So that's actually the positive side of stress. So when somebody tells you, oh, that cortisol is that bad hormone, challenge them. It's not. You need it. You want to be able to have a cortisol response when you're faced with a tiger or anything else, like snow on the ground and slippery conditions. When does it start to become bad then? We'll be talking a little bit about that. Everything in red is towards the bad side. Unpredictable stress, bad meaning it starts to affect our bodies. There's a risk for it to start to affect it in a more negative way. So unpredictable stress, in more higher intensity, prolonged stress, repeated stress, this is when it starts to go into the negative effects of stress on your body and your health and well-being. So bottom line, repeated and cumulative stress or chronic prolonged stress can induce these long-lasting changes in the brain that increase the risk for stress-related disorders, even when the stress is removed, okay? So think about this. If I ask the person next to you to start poking you with their pen, they'd do it, and you'd sort of say, stop it. But they wouldn't stop, and they just kept at it. And now they're poking at you even more strongly, okay? So intensity is going up, and you're like, stop it. Now they're glaring at you, and they're poking even more, to the point that the skin is starting. So what's going on here? You can tolerate some amount of stress. You can sort of deal with it. You can say, stop it. You're in control. The person could stop it, and hey, you handled it. That was fine. But now they're sort of poking you more. They're glaring at you. They start to say, I'm going to do something terrible to you. And there's going to be a bigger response. You're starting to move away from the intensity goes up. It becomes unpredictable. You never thought that person looked very nice. They're dressed well. They're here for a conference. They look pretty good, right? It's like, wait, what's going on over here? And then it's going on for a little bit longer. It's not going away. It becomes uncontrollable, and that's the situation where we start to have higher levels of a stress response, probably getting flooded. Okay, so that's just one little bitty example, very acute situation. No harm, that person will probably stop. There are many other people you can scream, and things will be under control again. Turns out there's a lot of chronic stress conditions that we don't feel like we have control over. They may continue, and really, there is a grand challenge right now, which has been going on for over the last decade, where we do not have cures for these chronic diseases. And the reason is that they are not single gene diseases. They are not diseases that you can have one cure, one reason, one cause for it, and then addressing that particular cause and fixing and curing the illness. These are multifactorial. And what's really interesting are that they're psychiatric disorders as well as medical disorders, and they're connected. They're very highly connected, and one way of thinking about their connection is that they're connected through the stress pathways. And the reason for that is that stress goes right from your brain to every part of your body, and we're going to see some of that. Okay, so this is the grand challenge, and there are all of these types of uncontrollable stressors: trauma, abuse, loneliness, violence, victimization, other uncontrollable events, poverty. If you stopped for a moment and thought about whether you knew somebody who had been a victim of violence or aggression, psychological or physical, we're going to be talking about what happened to their bodies and their brains when things like that start to happen. If they were a victim of trauma and abuse, these rates are pretty high. They are not 5 to 10%. It's bad enough if it's 5 to 10%, but we're talking higher levels, and you're going to see some of those numbers. So, of course, there are genes that do have an impact on these stress-related illnesses, and there's very good data out there on genetic influences for obesity, diabetes, hypertension, heart disease, mood disorders, depression, anxiety, all of that, alcoholism. However, they're not, like I said, single gene disorders that you could have a genetic predisposition. There are multiple genes that are involved, and that stressful life events and adversity can help really sort of pull the trigger for manifestation of illness. I heard a nice little metaphor, way of representing this, that you might, the genes are sort of loading the gun, But stressful environments and exposure to toxins and lifestyle factors are the things that pull the trigger for these kinds of chronic diseases. So that's a borrowed phrase, but it's a way of thinking about what happens, what's the underlying multifactorial mechanisms or causes that may be linked to these diseases. So what happens when stress piles up? We now have ways of really codifying and assessing people's, what I like to call stress load, or what's your background. We all have stressors. We've, in fact, faced them, dealt with them, maybe sometimes not so well, but perhaps overcome them. And there's a resilience to humans, and we'll talk about that. But we do have a stress load. And so one question is, how does that impact our brains and bodies? So we do these structured interviews. We ask you about major life events, abandonment, divorce, separation, loss of child, parental substance abuse, relationship difficulties, some of these I mentioned earlier, life traumas, losing your home, witnessing or being in an accident, being a victim of a physical, sexual, and emotional abuse, being shot at, assaulted, being tortured, being in combat, losing someone to violence, recent life events, some of these happening in the last 12 months or the last 6 months. And there are impacts of stressors if they are recent or in the past. So that's really important. And then really our own individual vulnerabilities. The genes come into this play, but really we handle stressors differently. We approach them differently. And so thinking about someone who may be feeling more easily overwhelmed than somebody else, maybe more emotionally reactive, and that's okay. The question is really, how are you going to handle that in that emotional reactivity? So chronic stress is a component of what we assess, and that's more the subjective, perceived component. The others are really ways of sort of thinking about codifying the types of events that have occurred in somebody's life. And so that way we can sort of come up with a stress load, or how many bad things have happened in your life. And across the room we can sort of get a sense of 4 for somebody, 8, 9, 10, 15. And we like to think of that as that's your stress load. Walking into any kind of situation, you have to think about your stress load. Why is this important? In basic science research, there is very good evidence that when there's cumulative or repeated stress, an animal that has faced multiple hits or repeated stressors compared to control animals, there are actually changes in brain cells as a function of that. So here's a control brain cell in the frontal part of the brain, and we're going to be breaking down the brain model very, very soon. But here's a control. Can everybody see that? The yellow dot in there is where the cell is, and then there are these branches in blue and red and yellow just to show that. That's one nerve cell. We have millions in our brain, okay? That's one nerve cell. So what happens to that one nerve cell from an animal that's faced repeated stress compared to an animal that hasn't faced repeated stress? What do you see in there? Anybody want to take a shout out? Yes. Brighter dot. Okay. But what else are you seeing? That's much less connection. Much less connection. So there's a single neuron. You think, so it seems like things have fallen off a little bit. If you think of this as a branch, it's sort of like the branch shaken up and things falling off. Maybe leaves falling off and things kind of shrinking. And in fact, that's exactly what happens. These things are called dendrites, and then there are on their connections are connectors called synapses that connect with another nerve cell. And those things start to change. And they start to fall off, they start to shrink down. And so if you were to think about this happening to one nerve cell, what happens if you start to have millions in a particular, there are thousands in one region, the thousands in another region, and they're starting to fall off. So if there was a ball here and things were falling off, what would happen to that ball? Would it start to kind of go down in volume if things are falling off? Maybe, right? Would you buy that as an explanation? Okay, so we now have brain imaging techniques in humans. We can actually say, well, do we lose our nerve cells or their branches even in humans if we've had repeated stress? It's a valid question. And how would you actually know that? How you'd know that is by looking at what we call structural morphometry, or really brain volume. And we're looking at gray matter volume, which is your nerve cell volume. And you can't look at one nerve cell in humans, obviously. We're looking at brain imaging, structural brain imaging. And don't look for things falling off here. All this is showing is that this is where the hot spot is. So we asked the question. We took 103 community adults ranging from the ages of 18 to 50. The mean age is about 27. And they did not have any other illnesses, but we codified them on cumulative load. What was your load? What was your load? And we were able to then say, all right, let's see if this load impacts brain volume and tell us whether brain volume increases or decreases. So we asked the question of the data, whole brain. These are the hot spots that we came up. So all that yellow blob is telling us is this is where the action is. This is where there's a reduction in volume as a function of cumulative load. And here's the paper. That same image is right here, and here's the distribution. So this is a correlation analysis. It's not a controlled experiment per se, but this is really the volume, the number of life events, so that's your load. As load goes up, the volume in this part of the region, which we call the prefrontal cortex, and here, which is the striatum, and I'm going to talk about those regions, and the insula, all shrunk down in volume, about 10 to 15%. Healthy folks. Now you could say, well, you know what? Maybe I'm okay with the 10 to 15% loss. I can still function, and that is absolutely true. Are you functioning optimally? That's our question. Can you do anything to recover these? And that's really the important piece. We do think that, in fact, the brain is very dynamic. There's beautiful data now from neuroscience that shows that these nerve cells, certainly their branches, can come back, and there are ways to regenerate or have what we call neurogenesis. So that's an important piece, but right now I just want you to ponder what is happening here, and we're going to talk about these brain regions in a minute. As you're looking at this, the nose is on this side, okay? So there's a hint. Just if you put your hand on your forehead, we're talking about the front part right here, okay? So we'll come back to that region. Okay, so we are right in about the brain now. And you're going to see a model right here. And you're going to give me some latitude, since I'm the speaker and you maybe have no choice. But this is what we're going to do. I want everybody to lift up your dominant hand and make a fist. Okay, and I'm going to go stand by this picture, and I'm going to put my fist over there and ask you to extend your mind and see if this represents, one portion of the brain represents what's in your hand. Okay, so if I do this right there, is that kind of like, maybe, kind of like mirroring the model? So right here, this is the head, this is the fist, and this part of my arm is coming down. Is that okay? Can you just kind of give me that latitude? Yes? Okay. What's coming down is your neck right here, your spine, right, coming down into your back. But it goes up into what we call the brainstem, and then into this fist, which is the more primitive part of the brain. That's where we have our emotional brain, our reward brain. Okay, if you think about that, there's the amygdala is in there, the hypothalamus is in there, which gets your body signals. And that's the brain, part of the brain that's reacting to stimuli coming at you. Danger, love, good, bad, all of that. And in charge of giving signals. This is not okay, this is okay. And then also, good things are happening. Reward, pleasure, that stuff. So that's in your fist. Now I want everybody to go back up and give it a cap. Okay, and here are all the folds, and that's your cap, right there. Your brain in your hands. So think about this. It's a very simple model, but it's a way of, you have physicians, cardiovascular physicians telling us about our hearts. Oh, there are these 4 chambers. Well, you all are here because you're thinking about psychology and mind and brain, and you want to be able to have a model in your hand of the brain and how to think about it, okay? So right there, fist, reward, pleasure, emotions, and then here's the cap. And the cap are these thinking brain, okay? Thoughtful, flexible control of behavior, attention, all of these different cognitive and cognitive emotional and regulation skills are in the cap. And partly there's this beautiful dance that goes on between the fist and the cap. And what you want to be able to do is to really have the cap working for you, because you want to be able to think, hey, stop poking me with that thing, and coming up with a plan, right? If you're feeling kind of droopy and sleepy, you may not address it. You might shift seats, and that's okay. That's a good strategy. But somebody else may come sit over there, and that person will start poking them again, right? So there are different choices. One is not good or bad. There are different strategies that we do for handling stressors. And the point is that the frontal part and the parts that are in your cap actually play a big role in figuring out what to do when you're faced with stress. And this is important because of our ability to really think and save ourselves under situations of distress. So there are all of these activities that are going on in the cap, regulating attention, inhibiting inappropriate actions, regulating emotion, as well as regulating our body responses, our hunger, and all of those things. And this region that is in blue is called the prefrontal cortex, right there. When I told you, remember, your forehead, that is the region. Does this make sense? That is the region where that yellow blob was? Does that make sense? So you're losing neurons in there with when people are facing more repeated stressors. And that's concerning to us. There's also some other regions in here, but that's a really important region, particularly for regulating emotions and bringing down emotionality. So what happens under stress? The fist gets up. This is why you want the model. The fist gets overactive. There's heightened state of alertness, but there's also, oh my gosh, I don't know what to do. We're figuring out, and the signals are just ongoing, just pushing. Something's got to be done. Alertness is in high gear. The downside of the cap is that after a little while, it can get in overload. When it says atrophy, it just means that it can get too overloaded and starts to droop. So if your cap starts to droop, you're not going to come up with very good ways of resolving the situations. Or you might start to withdraw. Well, you know what, I don't know. I give up. I'm helpless around this situation. A lot of times people do feel, and the more uncontrollable it is, you can get to that state pretty quickly. And what's happening is that the frontal systems are not working very well, or they just droop down. They're spent. Okay, so the great thing about having all these folds and having this wonderful cap for humans is that we have a lot more folds than other mammals. The downside is that it's not unlimited in its functioning. It starts to get saturated. Okay, it's limited in its ability to manage and regulate things, and it starts to get drooped down. There's a chance that, in fact, the greater the reactivity and longer, more intense and longer the emotional reactivity and the drive from the fist part, the more quickly the frontal systems will droop down and your cap will droop down. Okay, so that's the model to be thinking about. So you get stressed. How do we know we are stressed? Yes? Your heart is beating faster, you start to get tensed up. Maybe you notice, maybe you don't. Maybe you start running, you want to get away from the situation. You get up and move from the person who's poking you. You do that immediately. I'm not going to be over here. We react, we use nonverbal and other ways of taking care of situations, verbal as well as nonverbal. So we have these sensations, we express ourselves. There's the fight or flight response, which you all have heard. And then there's the hormones and the immune system that sort of go into high gear to mobilize our resources. If we had to run, you actually need more energy. So in fact, cortisol and adrenaline play a very important role in signaling the muscles and rest of the body, signaling glucose. You will see some slides on that. Cortisol directly signals glucose storage as well as other pathways to really give us the energy to think and to move. So in an acute situation, we do pretty well. And in fact, how many people have heard of good stress and bad stress? Oh, this is good, right? People hear about this all the time. It's all over the internet. Oh, stress is good for you. Oh, no, stress is bad for you. What is that about? Well, in fact, stress is crucial for adaptation. You've heard me say that, but really at mild to moderate levels. If 5 of you were together waiting to, you had a 3 p.m. deadline to turn in a project. You're working together. There's a lot of team work going on, you're alert, you're aware, there's increased energy, there's focused attention. your working memory is going well, cognitive ability is all driven, everyone's talking, your performance is enhanced, and you're going to make it. And then you get a phone call. So right here, mild to moderate levels, the juices are flowing, adrenal cortex is secreting cortisol, epinephrine, you're pumped up, the team is working really well. You get a phone call. Guess what? The deadline is 1 p.m. Whoa, what just happened? Did it move from mild to moderate to too much? There's no way we're going to do this. That's it. One person says, all right, I'm done. Throw something out of here. The other person has a different reaction. A third person has, and one person is left saying, no, wait, we can actually do this. How many of you have been in situations like that? Yes? Are you connecting with this? Right. So you're moving from mild to moderate towards the uncontrollable zone. There's no way we're going to have our write-up done by 1 p.m. 3 p.m., yes, it was all planned out, it was done, manageable. So now you go into overdrive. Right here, you see that? Here is the amygdala and that fist part, and that's in overdrive. But in mild to moderate levels, you've got all this going on. It's working well. And that prefrontal cortex is in yellow and red, which means it's also hyperactivated. It's managing. There's a nice dance going on between the fist and the cap. The moment you start to go into the other side, this starts to droop down, starts to turn blue. In our way of thinking about brain imaging, it starts to turn blue, meaning go hypoactive, start to droop down, and the amygdala and other parts of the brain, the reactive brain, are saying, we're done, we can't deal with this. Okay? So the prefrontal cortex, which we talked about, which is in your cap, and it links to the other cortical regions, are involved in cognitive, planning, problem solving, goal setting. All that was going on in that example. There are some things that are not so great in coping. These are active coping skills. These are more avoidant coping skills. Then there's the biological response, which we just illustrated. And then you could have behavioral responses. How about in that situation, somebody said, you know what, let's just take a 5-minute break. We'll come back. And everybody takes a walk. Anybody done that when you're feeling overwhelmed, right? You just go out, you take a walk. That's a behavioral way of regulating stress. You come back, say, you know, I just cleared my head. We're going to call them and say, listen, you can't do this to us. Wow, came up with a thought that actually, you know, is a way of problem solving and planning, right? So that can happen. Those are positive behavioral coping responses. And then there are things of sort of, we're done, let's go down to the bar. We've failed on this one. How many people have done that, right? We're done. We're not going to make it for this one. We're going to have to wait for that next project. So you're at the bar. Okay. So what happens to our brain when we're in that moment of getting flooded in? You're feeling really stressed out. We've been able to, I showed you structural changes in the brain when you've had a history of stress. Now I'm going to ask you to think about, we're going to stress you in the scanner and what are the changes happening in the brain. We've been able to actually stress people for just 5 minutes, so that's not the same as feeling really, really uncontrolled, but we have them think about really uncontrollable situations. And what's in yellow and red here are the regions that are very overactive, okay? And I'm just going to cut through, this is which is amygdala, hippocampus, striatum, which is our reward regions. Those are all things in the fist. Everything in yellow, red here is the fist. So when they're feeling really stressed out, you start to see overactivity in the amygdala and the striatum and in regions of the fist. So I wasn't just being funny about the model. It actually ties right to the data here. And then what's in blue here are regions that are getting droopy and not as functional. So they're in blue, meaning they're deactivated, and that's the frontal region. And so under brief stress conditions, and we check and make sure people are stressed and they give us ratings and all that, you see this kind of dynamic. Droop down and upregulation of the fist, of the reward regions and the limbic regions. And you might say, well, that's people imagining their stressful situations. We used a different methodology to provoke stress. And once again, the same regions were activated. Okay, once again, the fist was overactive, and these blue regions are these frontal parts of the brain right here behind your forehead. and that was deactivated, and those are the ratings on a scale from 0 to 10. People were at a 6 or 7 during this brief period, okay? So these are dynamic changes that are going on under high levels of stress. And in fact, then we were able to show that those blue regions in the prefrontal cortex are related to the hippocampus, which is in your limbic regions, and then that dynamic, so lower activity in the prefrontal cortex and higher activity in the limbic regions predict a scale that we use called the Cornell Medical Index, which is an index of how many physical and psychological symptoms you're feeling right now. So those are just symptoms of distress that the body and the mind may feel, and we're going to come back to that. So that was predicted by the brain response, okay? So how we feel when we are under stress actually has something to do with these brain changes that we're showing you in controlled experiments, okay? So here's one model, one way of thinking about it, and we're going to transition to childhood because, like I said, stress doesn't start today or yesterday. We live long enough and we have long childhoods, and people face all kinds of challenges through childhood today. And so one way of thinking about it is that if you have low stress, you have good growth in your prefrontal systems and your cortical systems, and those then control these limbic fist regions over here, and you have good adaptation. You can manage reward. You might say, gee, going to that bar would be nice, but maybe we could try to finish that project and then go to the bar. So that would be good regulation, more controlled use of things that are adversarial to health, and you have better health. Whereas under high stress conditions, this is sort of a simple model, you have droopiness or atrophy in the prefrontal development, and the fist is overreactive, and you have impaired behavioral control, and perhaps more risk for chronic diseases. So this is a model that Hilary Blumberg and Linda Mays and I came up with at the Child Study Center, and we've tested it in children and in adolescents. Now, before I show you some of that data, let me just show you some epidemiologic data on adverse childhood experiences. These are the ones that the CDC records, and red and yellow are just showing in measurements that were done in 1998 and then 2009. And you see that by 2009, these are different samples, so it's not a direct increase, but just ways of thinking about in the population, in the U.S. population, there seems to be some increases in the number of adversity that children are facing. And right here, when it says ACEs, that's adverse childhood experiences, you see that by 2009, there's a decrease in the number of kids who have not faced any adversity and an increase in those who have had 4 or more. Why do we care? When you look at odds ratios over time, longitudinally, there is a direct relationship between the number of adverse childhood events you've had in children as a child and then occurrence of these different health conditions in adulthood. Depression, ischemic heart disease, cancer, stroke, bronchitis, emphysema, diabetes, and obesity. Okay, so this is important. Stressors during childhood, while these stress pathways are developing, are very critical to shaping how we cope with stress and also how much of our CAP fully develops and how that then feeds into our health pathways that are driving positive versus negative health. Here are data from Jane Taylor's lab and Dr. Koleski and Ralph DeLeon. And this is really, I'm going to run through this very quickly. They gave adolescent animals, cortisol and infusion of cortisol continuously for a period of time and showed that the longer, so if you have high levels of cortisol and sustained, that's not good. Acute levels is great. It helps alert us, helps us manage stress. High levels sustained is not good, and what it led to was increases in habitual alcohol intake, stress-induced errors in cognitive tasks like making impulsive choices and things like that, and there's a paper that has come out of that. This is an actual neuron, and what you see from the prefrontal cortex, and you see more stubbiness, so what I've already shown you, which is those dendrites and branches falling off, and this is actually showing that there's with corticosterone, high levels of corticosterone, there's more stubbiness, and atrophy of those dendrites, and then some genes that start to have an impact on prefrontal cortical neurons. In a brain scan, when you scan adolescents, structural brain, there's lower brain volume in that frontal region of the brain behind the forehead. There is, with higher levels of stress, there's lower activation of that frontal cortex. It's related to how many errors we make on cognitive tasks. There are some sex differences in the way stress affects those prefrontal neurons and circuitry that would be helpful in regulating stress, and so on. And then it starts to predict who engages in substance use behaviors in adolescence in a longitudinal study. So again, these circuits are having a really critical impact if they are faced by stressors, and then tracking the behaviors and that may feed into our ability to cope with stress and regulate ourselves. So just to summarize, experimental stress exposure impairs attention, sustained attention, working memory, cognitive flexibility, goal-directed behaviors, increasing habits, decreasing goal-directed behaviors, increasing impulsivity and risk-taking. And those, again, those abilities are important in our ability to regulate stress. And of course, these are some behaviors or choices we may make under stressful conditions, and these are the top 3 behavioral contributors to chronic diseases. Smoking, binge-heavy drinking, overeating, poor diet are all impacting those chronic disease conditions that I was talking about. It turns out that actually having number of stressors or stressful life events, this is a study by Laut, and they were looking at gene stress interactions, but really what I want to show you is even with different polymorphisms or different versions of this particular serotonin transporter gene, which each of us have, we might have one or the other of these. Those who have high stressors in their life, this is a longitudinal study of children in Europe, following them right from birth, and then looking at their binge drinking at age 19, 20, and 21. Those who had high levels of stress were just consuming more alcohol. Okay, longitudinal study. All right, what about food? This is really closer to home for all of us. We need food to survive. We're all eating. We love some of the foods that are out there, the fast foods and rewarding foods like cupcakes and chocolate and candy and chicken wings and pizza and all of that. Anyway, under stress, there's a really interesting phenomena that people have noticed. About 40% will increase eating under stress. About 30 to 40 will decrease, and about 20 will have no change, okay? Lots of studies have shown that. And in fact, here's that brain model again. Once again, your fist over here, your cap over there. There's that part behind your forehead, the prefrontal cortex. And high-calorie foods stimulate things in the fist, okay? And the prefrontal cortex has to regulate the reward regions and also the emotional regions and the regions that are regulating or impacted by our viscera or hunger signals. And so that's motivation and reward pathways. All right, dopamine, how many people have heard of dopamine? The reward pleasure signal, it's not just the pleasure signal, okay? The mass media likes to talk about it that way, but it is actually really important in learning and memory. When you are in a team working together and trying to figure out, solve a problem, dopamine is playing a role. You're coming up with new strategies, dopamine is playing a role, okay? So it's important in motivation and in learning and memory. And so these dopaminergic agents, really interestingly, are actually also important and have an influence on appetite and food. So food is one way of thinking about another addictive substance or rewarding substance, right? And so medications that block dopamine can have an impact on weight. Medications that inhibit these dopamine receptors, those that block the reuptake of dopamine or increase dopamine, can decrease weight, and those that inhibit the dopamine transmission can increase weight. So anyway, what you will want to notice now is that if I put threat and stress signals, the same or very closely overlapping circuitry is activated. So these reward and stress circuitry are very tightly connected, and both are involved in the prefrontal cortex in regulation, okay? And these are the molecules, the stress molecules we talked about. I'm going to let your professors tell you more about these, but just to give you a signal that these are all important in regulating stress, a number of them actually are involved in addiction as well as in food intake. Okay, so I'm going to show you a study. In the interest of time, I guess I'm going to perhaps skip this slide and go to a study that we did with our endocrinology colleagues, where we manipulated the level of glucose in our body. Okay, we want glucose levels to be steady. That would be a euglycemic phase, written right here. And so we did an experiment in the brain scan where we kept glucose levels standard at the normal levels, around 88, 90. And then we manipulated hormones in such a way so that your glucose levels drop down to about 68. That's mild hypoglycemia. You don't feel any clinical symptoms. You're not ready to faint or anything, but glucose levels are going down. So you would expect at that point that the body is going to mobilize, right? There's now an internal stressor happening, which is your glucose levels are going down. It will do whatever it can to survive and protect ourselves under those conditions, because you don't want to go into the worst case scenario, which would be a coma or drop down. So you don't want an emergent situation. And then what we did while we dropped this, kept it at normal levels and dropped them, we showed people pictures of ice cream, cupcakes, cake, chicken wings, pizza, some broccoli and tofu and carrots, and then some chairs, tables, lamps, things like that. Okay, and for each picture we asked you, how much do you like that and how much do you want it? All right, because what we wanted to do, what does the brain do when you start to drop glucose levels, right? The brain uses the most amount of glucose as any other organ system, and that frontal part soaks in a lot of it. And in fact, when glucose levels dropped, cortisol started to go up. It's like, whoa, internal state not doing well, let's start mobilizing. So cortisol levels started to go up. We had a control condition where we did not drop anybody's glucose, and there was no difference in cortisol over there. Very quickly, the bottom line, just in the interest of time, I'm going to show you what the findings were, okay? So firstly, there was an increase in liking and wanting of high calorie density foods or high fat foods. Okay, so we just liked and preferred the cones and cupcakes more so in the hypoglycemic phase compared to the euglycemic phase. Same person under those two conditions, controlled experiment. Okay, what was happening in the brain? There was greater activation. Everything in blue here is actually greater activation. And this is a different orientation, but take it from me, this is where your fist is, and that was in overdrive. So everything in the limbic regions, emotional motivation regions was in overdrive. What's in red here is your prefrontal cortex, and that was drooping down. It's like, okay, shut that down. We don't need to be thinking about whether we should be eating or not. We need to eat. We need to figure out what to eat. And we need to eat. We start wanting more of things that are going to bring glucose levels up really quickly. Okay? So our motivation, our preference all shifts toward increasing caloric intake. And that can be a scenario when you're highly stressed, either from stimuli outside the environment or from stimuli inside the environment. And then, in fact, glucose levels, lower glucose levels were correlating with activation in the prefrontal cortex. Okay, so when it's higher, under normal conditions, you have greater activation. When it's lower, you have deactivation. And cortisol was correlated with levels in that fist region and in the motivation regions and driving us to go get some food. All right, so stress can influence eating behavior. Particularly, it'll shift us towards eating snacks, fast foods, calorie-dense foods, highly palatable foods that may, in fact, get glucose levels up, but we might also overshoot, overeat, and then we can have a perfect storm around that. Again, in the interest of time, this is a complex little diagram here, but what I want you to know is right there, once again, is a picture of the brain, the fist, and the spine coming down, your spinal cord with the nerves there, and then there's your frontal part of the brain. And it's another way of saying that these regions are actually regulating what's happening in the rest of the body, including the immune responses, It's just a good thing to note that there's a tightly complex, tightly connected system between the body, immune responses, cellular changes as a function of stress, and how the brain is controlling and regulating that. Okay, so how do we know we are stressed? We're back to that slide. We might have cognitive symptoms, we might have emotional symptoms, behavioral symptoms, physical symptoms. Not all of you will have the same ones. We're pretty individual in where we feel our stress and how we express it, but these are often the ways that we think about it. So we like to think of this as stress signs and signals. You might say, do you have to scan my brain to know whether I'm losing those neurons and those branches? I'm going to say, well, it would be good, and we don't have a test yet to see whether your load is bad enough that it's affected your brain. But you know what? At the Stress Center, we like to say, how many of these are you feeling right now? And if you have 3, 4, 5 or more of these, and you might start checking them off right now in your mind, then your body's starting to feel some stress. And I would say pay attention to it and start thinking about what we can do about it. So what can we do about it? We've been doing some work with medications development to try to see whether there are actually medications that can be developed to target this particular circuitry that I've been talking about, rather than just having a medication for obesity or alcoholism or mood or anxiety. How about we start looking at the mechanisms that are driving these states, these pathological states, and begin to see if we can reverse the pattern in the brain there, reverse the control centers in the brain. Okay, so we've tested a couple of these medications. I'm going to show you about that, and we didn't dream this up on our own. Amy Arnsten at Yale, who does non-human primate work, came and looks at the prefrontal cortex, showed these pathways, the cyclic AMP and protein kinase C pathways are seriously affected by stress, and they can be blocked by guanfacine, prazosin. I'm going to tell you a little bit about them very quickly. These are agents that have been around for a long time. They reduce blood pressure and first came out as blood pressure agents, but turns out they have central effects, meaning they affect the brain and they have effects on the prefrontal cortex and tend to rescue those brain cells in the frontal part of the brain, okay? And so there's something like prazosin, which is an old antihypertensive drug. Turns out it rescues stress-related impairment in the frontal region of the brain. It decreases PTSD clinical symptoms. It decreases drinking in alcohol-dependent individuals and in rats. And it decreases stress-related alcohol craving and anxiety. Very quickly, here's some data on that. Prazosin improving dream ratings, decreasing PTSD clinical symptoms. That's the rating for clinical symptoms. PLA means placebo versus prazosin. And baseline and end of treatment. So basically, from baseline to end of treatment, there was a real decrease in PTSD clinical symptoms in a clinical trial with prazosin. So prazosin is supposed to improve sleep and actually decrease trauma-related activation. This has been tested in veterans and other groups that have PTSD. And we looked at prazosin after prazosin versus placebo in a controlled experiment. We showed decreases in alcohol craving among alcohol-dependent individuals, decreases in anxiety, decreases in negative emotion, and we're doing a phase 2 trial really to see if it actually can have sustained impact on alcoholism as well as on stress and anxiety in those recovering alcoholics. Guanfacine is another drug that actually inhibits adrenaline in the brain. Too much adrenaline in the brain can not be good for our frontal regions of the brain. And all this is showing, I'm going to rush through this slide very quickly. Under the Stroop, with doing the Stroop task in the scanner, we have a reduction in activity in the frontal region of the brain, but if you do have guanfacine, you actually increase that, which is right here in this frontal region of the brain, as well as a couple of other regions. So that's another neurochemical and a drug that is around and available and may actually improve frontal functioning under stressful or challenge conditions. There are, of course, behavioral strategies that we can use, and we're using this at the Stress Center. You heard that we now have clinical services. We also do prevention and wellness services, and we use a lot of these techniques because they, in fact, have been shown to have an impact on that brain circuitry. I'm going to end with one last slide, which shows that impact. So this is a slide of what is mindfulness, one before the last. How many people have heard of mindfulness and mindfulness training? Fantastic, great. So there's a whole self-help movement about mindfulness, but there's also a growing tradition of psychotherapy where mindfulness strategies are used, but really in the mold of psychotherapy and in talk therapy, and we do that in different ways. And it's different from the way it's implemented in the self-help tradition, okay? But some of the important components is paying attention in the moment, non-judgmentally, being in being mode as opposed to doing, getting up and leaving. I'm giving up. How about we just sit with it for a little while? That would be the being mode. Observing, letting go. These are all aspects of what we call mindfulness. And I'm going to show you a slide where Judd Brewer, who was one of our faculty at Yale, and he's moved now to Worcester, Massachusetts, and he did a study in collaboration with us where we looked at 6 weeks of mindfulness training versus a psychoeducational don't smoke, smoking is bad, with smokers who wanted to give up smoking. So they could either mindfulness training or freedom from smoking psychoeducation. And what we found is that after mindfulness training, the fist, right here, the amygdala, but also the insula and striatum, the areas in the fist that are involved in emotional reactivity and in drive and reward drive were, in blue it shows decreased in activation, okay? After 6 weeks of mindfulness training. And that was more so in what's MT, which is mindfulness training. It was lower there for amygdala. And then that predicted and was related to how much reduction in cigarette smoking they had, both at the end of treatment and then at follow-up. So once again, doing behavioral interventions can have an impact on brain response and on this target circuitry that is very critical for alerting us about what's happening under stress, alerting our bodies to do things to adapt and cope with stress, whether it's good or bad, and then also giving us the regulation abilities from the circuitry in the cap. So with that, I want to thank you for your attention. I want to show you that at the Stress Center, we do have clinical services. Hopefully the science of what we're doing there gets translated immediately and more thoughtfully in an integrative manner to treat the whole person. And these are all of the different types of services we do have. Thanks so much.
Thank you, Dr. Sinha. I think we have time for one or two questions, and then before we do that, I'd like to make a quick announcement. There's one room change for the tweaking brain cells, electroporation techniques and applications. That's now room 227. It's directly across the hall from 224. So you won't, if you can find 224, you'll find 227. So this is working. So does anyone want to ask Dr. Sinha a question or two? I see a hand over here.
Hi. Sorry, can you hear me? Yeah.
Hi.
Closer? Okay. In your hypoglycemia studies where you were doing the scan and the stressing on hypoglycemia, how did individuals that under stress do not eat, how do they react to those images?
So we actually, in that study, we were not looking for people who don't eat versus those who eat. We were really asking the question, if you have a physiologic stressor, like mild hypoglycemia, what does the brain do? And is there a shift in preference and in motivation for foods, and particularly high-calorie or high-fat foods that would then be the signal for eating and preference for those foods, but eating those more and perhaps eating more of it. So it's a very interesting question that you have. We're quite taken up with that, and we are now conducting a larger scale study where we're bringing people into the laboratory, we're stressing them out, and we're looking at who eats under those conditions. So we're actually presenting, it's an experimental study. We're presenting them with foods, a snack food. And if any of you have been in that study, I shouldn't be telling you the hypotheses, but anyway, it's a study that does start to point to sorting out individual differences for us. Well, I don't have the results yet.
Okay, thanks. So we'll take one more. Can you introduce yourself too?
Hi, I'm Prakriti. I'm from Mount Holyoke College. So I wanted to ask you a question about like corticolimbic connectivity, as you talked about. So you had talked about heightened limbic activation in high stress situations and relating to atrophy in the prefrontal cortex. Now, I guess what I'm confused about is if this is a state of, internal versus external stress. You could think of how they sort of feed into each other. So do you have any, can you speak a little bit more to, in, say, an internal stress situation, you have heightened limbic activity, which decreases prefrontal activity, or like the other way around, and when they flip, if they flip, that kind of thing?
Right. It's a great question, and maybe I'll just expand a little bit on it, because I think perhaps this is what you were thinking about, which is we may actually have scenarios where the body is stressed out. For example, physiologic conditions like obesity or diabetes, or being just stop drinking alcohol and getting past acute withdrawal. We do a lot of studies with those who are wanting to give up alcohol, and even after acute withdrawal, the body hasn't fully recovered. So that would be an internal stressor. Now we don't lock ourselves up in those scenarios. We're dealing with the world, and you can have stress during that period of time. And so we've asked the question: what happens? Are those people more vulnerable to stress? And perhaps that's related to your question, which is the interaction of these states. We like to think, just model-wise, we like to think of those conditions where there are stress-related adaptations in the brain and the body. And then this heightened limbic reactivity and decreased prefrontal reactivity is not necessarily the model we'll see, because there's some adaptation going on. And we've actually published on this, so I can give you some references. You start to see different patterns. So for example, in the recovering alcoholic brain, 4 weeks abstinent, I took out that slide in the interest of time, you will see during basal or relaxed states a hyperactivity, not just in the fist part of the brain or in the reward region, but also in the prefrontal cortex. Sort of at baseline, it's like this. It's all hyperactive. And when I stress somebody, that's under relaxed conditions. If you and I were relaxed, the prefrontal cortex actually would take a break, and everything would sort of take a break. Because it wants to be mobilized when you're under stress. When you stress a recovering alcoholic and look at their brain, what happens is that the prefrontal cortex doesn't turn on. And the control person's brain will turn on, at least initially, and want to start to regulate the fist, right? But the alcoholic brain is not going to turn on, and there's a lot of blunting. So it's as if the juice isn't there. These circuits that have to help us regulate and help us cope and survive are out of commission. So there's for different conditions, it comes up differently. It's a really good question, and we have to sort of go through the tea leaves, look at each condition, and see what's the impact. Because ultimately, it's affecting the person's ability to manage those situations and handle them and really be resilient and be adaptive under stress. And in fact, we know that they're at high risk for not being resilient, and we've shown that brain pattern actually predicts how quickly they relapse. So that would be a non-resilient clinical outcome.
Thank you again. Sure. So hopefully Dr. Sinha can stick around for a couple minutes if some of you have additional questions. Can you please join me again in thanking her for such an engaging talk?